We'd hit that 95% threshold. We stopped the trial. And so that was a good result. We were happy. But as my colleagues were probably initially happier than me as an annoying statistician, wanted to try and think of all the reasons why this might be wrong.
In this episode, I talked with Dr. David Macleod. He's a statistician at the London School of Hygiene and Tropical Medicine. He has this interesting paper with colleagues in Kenya about an app called Peek Acuity. And what it does is it does screening for eye care issues. Now, what they found is they could screen people, they could find eye care issues that did need to be resolved. But when they referred people to further eye care, only about half, just under half of people followed through with the referral. And it was only about a third among young adults that needed eye care. So what they did was they added a condition where they could give some motivational counseling. The person doing the screening could give some motivational counseling to say, hey, you have these eye issues. Here's a referral, and here's why you should go get it. And they decided to embed a trial within the app. So randomly, a notification triggered to the person doing the screening to give this additional counseling. And they could have chosen to do a standard clinical trial, right? Figure out a right sample size, recruit that number of patients, and then do the randomization and implement and see what the difference is. But instead, they chose to do a Bayesian adaptive trial. So what happened is every week they were looking at the data, updating their prior beliefs, and stopping the trial when they reached a certain threshold of certainty around whether the intervention was effective. Dave and I talk about this trial design and its pros and its cons, and I hope you enjoy our conversation.
We're going to talk about interesting methods today, but what's the eye care challenge in Kenya and what's the motivation for this study?
It started quite a long time ago, before I was ever involved in it. One of my colleagues, an ophthalmologist called Andrew Bastavris, he was doing his PhD project in rural Kenya, doing eye tests for population there. And he had a big ophthalmoscope sitting on the back of a truck going down rural roads. It was a big, expensive piece of kit, quite hard to move around, and also eye charts, the type you would normally get in your local eye doctor's office. Setting those up was a bit more complicated, making sure the light was right. And he thought there had to be a better way. So he's developed eye screening app, testing for visual issues, just an eye test, using a phone app. And that's been used to screen people for eye problems across the world in other different countries. Alongside that, he's built a huge bit of infrastructure around data collection that they use within their programs to identify areas for improvement. They have very good data. When someone gets an eye test, that data is collected. So once someone is identified with an eye problem, they get referred on to further care. They can then track whether that person has attended the next stage in the process. Only about less than half of those people are actually reaching the next stage. So we're finding people who need care, but we don't think they're accessing the care that's needed. Part of the program of work that we embarked on was trying to find out the reasons why. We collected some prospective data to try and identify who were the people that weren't attending. What's the characteristics that those people share? We looked at where they lived, gender, age, the type of eye problems they had. And so then that helped us identify which groups of people were less likely to seek the care that they needed or were unable to seek that care. And then from that, we were then able to do some qualitative interviews and asked people, hey, you attended our eye screening, you didn't attend the clinic that you were referred to. Did you go somewhere else? Were you unable to? What things motivated you to not go? What would have motivated you to go? That information allowed us to come up with ideas in conjunction with the communities that we were working in. What might have helped you attend those clinics? And one of the things was a greater understanding of what the benefits would have been. Other things are cost of travel. Other things were the clinics being too busy. You attend the clinic and there was a long queue, then people wouldn't wait. And some of those problems are harder to address than others, require a lot more resources. But the suggested solution of providing a little bit of more information and some additional reminders was actually a very cheap intervention that we felt that we could apply quickly and was suitable for short, rapid assessment of whether it was effective or not.
Essentially some formative qualitative work. Those familiar with product design might call that design thinking or human-centered design. So you add some basic counseling to the app at the point of screening. Someone's identified as needing referral for eye care, and the intervention is a short, like you said, low cost, quick to implement intervention where you tell someone about what they can expect and why they should follow through with the eye care results. If we want to test how this works, we could design an experiment where some people get a version of the app where there's this counseling and other people don't. Then we'd have to go out and recruit people, and we'll set a sample size of, 1,000 people, and we'll randomize them. But you guys took a different approach, and one, embedding this directly in the app, which I think was a great opportunity, and two, doing a Bayesian adaptive trial instead of a traditional RCT. Talk us through what it is and why did you choose it?
Yeah, so the first part, embedding the research in the program, was a real fundamental thing that we wanted to do. So this was Andrew's vision. This trial wasn't the end point of it. We didn't just want to answer this question, does this additional counselling and reminder message improve attendance? We wanted to use it as almost a pilot for Peek. When we don't know the best thing to do, how can we set up a system that is fairly light touch, that doesn't require specific clinical trials teams to come in and work on it, when it can be done fairly simply in embedding it within the system. And then the choice of the adaptive design versus fixed design was, like in any trial, you want the answer as soon as possible. You don't want to have to wait too long, but you also want the correct answer. And the trade-off between those two things, that the longer you run the trial for in a standard trial with a fixed sample size, the better your answer is going to be, the more likely you are to get an estimate of the effect of it closer to the true effect. And I think this is where there's a bit of tension here. Maybe in a programmatic setting, what would tend to happen would be, we've got an idea for this thing we should do, let's do it. And maybe it would go in untested. And so you've got two extremes there. One is, hey, we think this would help, let's do it. The other end of the spectrum, which is a full clinical trial, a long duration to get the estimated effect of our small intervention. So we felt the ability to either discard something we don't think work well quickly, or if something is working very well, then we can adopt it sooner.
There's a clear rationale for doing this. How does a Bayesian adaptive trial work in practice?
We had a clear outcome, a binary outcome. Did someone attend their referral or not attend their referral? And really what we were looking was, as we progressed through the trial, at each stage, once a week, we were taking a look, and we were looking at, okay, how many people have attended the clinic in the group who were randomized to get the enhanced counselling, the additional reminders, and those that were not. At the simplest method, you're comparing those two proportions. Using a Bayesian framework also means that rather than comparing those two proportions and calculating a p-value, instead of doing that, we add this thing called a prior, which tells us about our prior belief as to how the intervention might work. We used a very, what we'd call an uninformative prior, which was that we allowed our prior belief to say, hey, we don't know how effective the intervention is going to be. It might be terrible or it might be great. And so we allowed this very much flat playing field of, we don't know what the intervention is going to do. Let the data tell the story. What I probably didn't appreciate then that I do now. I should also say, my colleague Min Kim is the person who did most of this work, who's on maternity leave, so she should be the one credited for this. She did this as her PhD work, and it's great. So what Min and I probably both didn't quite recognize was, I think our understanding, and probably a slight misunderstanding, was we don't really know how this intervention is going to work. So applying this uninformative prior is the safe option. We're not making any claim about what it does. And actually what that's sort of does is you're saying, oh, it's just as plausible for there to be no effect as there is this enormous effect. And that's not true. grew really, it's very unlikely to be a really enormous effect. We weren't going to get everyone attending the clinic afterwards. So we used this uninformed prior, which meant that the data drove our results. And that meant we came to a conclusion quite quickly. And we drew this conclusion and we make this claim that we were 98% sure that the intervention is better than not doing the intervention, because we've combined this prior belief, which is anything is possible, and the data, which had a higher proportion attending the intervention arm than the control arm. So what we did was we checked each week to see the difference between the control and the intervention arm. Each stage, every week, we made an estimate of how likely it was that the intervention arm was better than the control arm. And our plan was to stop the trial when we were 95% sure that the intervention arm was better than the control arm.
This is a key difference, I think, for folks listening who maybe are more familiar with a frequentist design, where instead of looking at the probability that your intervention is better, in a frequentist perspective, whether you know it or not, you're looking at the probability of observing the data, given that the null hypothesis of no effect is true, right? So we're thinking about probability in a very different way here. And that's why you're incorporating this idea of a prior and looking at the data. So what you're saying is you had this very flat prior saying really anything could happen. There could be a massive effect of, maybe this app gets 100% of people on a referral, or it could be that no one goes. I appreciate your perspective on this in hindsight, because if you look at the effectiveness of digital interventions just as a whole, they tend to have small effects. And theoretically, you could look at this as the intervention is really just, it's designed to be a brief sort of motivational counseling script. So we probably wouldn't expect the effect size to be massive from this, but it doesn't need to be massive to be useful and valuable. I don't want to put words in your mouth, but if you were to design it again, are you saying that you would put a narrower prior, a stronger prior that's maybe is centered around zero, but allowing there to be a smallish effect on either side?
Yeah. So I think probably if we were to do it again, we would suggest a sort of a more modest prior, a more stricter prior, or whatever that says it's probably unlikely to be making a massive effect. I think if I was to draw what I really believe the prior effect would be, I think it would be quite unlikely that it would have had a harmful effect. When you're doing a trial, you don't really want to make this claim about, we think our interventions, we're almost forcing it to be to one direction. But the reality was that I'd be surprised if this had a negative effect. So you'd almost want an asymmetric prior, and then I wouldn't really know what to do with that. I think if I was really doing it, I would put a stricter prior than we had put. You have a trade-off then, because you're going to run the trial for long if your fixed probability at which you are going to stop the trial stays the same. Everything's a trade-off. All we're talking about here is trying to stop false positive results. We could use a stricter prior to stop a false positive result, claiming that the intervention works when it doesn't. And we can do that by putting a stricter prior on, or we could do it by setting the threshold higher that we wanted, or there's various other ways we could do it. I think you've listed it in your notes, which are a really good summary of it. But putting a stricter prior seems to be the most realistic one. That's the one that kind of makes me feel like that's the true thing that is changing. Actually, our original estimate of what it could have been wasn't correct. We used too wide of a prior, really. So then our value of 98% probability we came out at the end is probably greater than the truth.
What's unique to the Bayesian approach is you can set this prior, right? And you obviously had some choice. And an analyst has the ability to set a prior based on their belief, and we could disagree on what the prior is. I think that's why some people don't like this Bayesian approach, is that it seems subjective in how you can do this. You set this prior, and then the data come in. And what it allows you to do in your approach, this Bayesian adaptive trial, is check. I think you said every 7 days as the data come in, and you're going to update your prior belief based on the data that you're seeing, which is very different than in a frequentist trial where, you can check the data at some pre-specified points, but you're going to pay for it in the alpha level you use. It's a much harder approach. In the frequentist approach, you don't just look at your data and stop when you see a p-value you like. But in your approach, you're able to look as the data come in and you're updating your belief based on the data. And you, I think it's set, some thresholds to say that we're going to stop when it's, 90% chance of being superior. Or I think you have an equivalency there as well. If it has a very high chance of having no effect, you'd also stop. Is that right?
So we had actually quite a lax threshold for the superiority and a very stringent threshold for equivalence. So to achieve equivalence, the trial would have had to run for quite a long time. And so you could have set a more lax threshold for equivalence. And that's what, when I mentioned at the start, is the benefits of these adaptive trials are, one, if it's really working well, you can stop it and then you can adopt it. Or two, you can discard early interventions that are not that effective. And in order to do that second one, you would have to set a sort of equivalence threshold probably more lax than we had. So we were quite well set up to catch an effect early, but we probably weren't so set up to discard an ineffective intervention sooner. And I think the decision about where those thresholds lie probably depend on the nature of the thing you are testing, the intervention. It's a very expensive intervention. There's got to be overwhelming evidence that it works, or the effect size is big for it really to be cost effective. Whereas if it's going to be quite close to zero, you might just be, if we didn't know that sooner, then we can discard that.
Well, this is why people don't like statisticians and methodologists, right? We always say that it depends. And it does, right? It depends on, like you said, if this is a really expensive intervention, you might want to optimize for a different answer. And so after 30 days, you stop the trial and find that the referral completion percentage is about 7 percentage points different between your arms, 32 among the group that did not get this counseling and 39% among the group that did. So a 7 percentage point difference, and it met your threshold of looking like it was highly likely to be your intervention. Tell us about that.
Bigger effect than anticipated. So we'd hit that 95% threshold. We stopped the trial, and so that was a good result. We were happy, but as my colleagues were probably initially happier than me as an annoying statistician, wanted to try and think of all the reasons why this might be wrong. And I think one of the things that we wrote about in this methodology was the high potential for a false positive result. Because of that uninformative prior, the potential for a false positive result was over 30%. That's if the intervention was no more effective, we would get a chance of finding that the intervention was severe almost 1 in a few times. So that was really quite large. But we thought that was acceptable because the intervention was so low cost, it was no risk. It's not like you're giving someone a new drug that might have other side effects. All we're doing is giving them an extra text message reminder and a bit of extra information. So we weren't so worried about that high type 1 error rate. But things that gave me confidence that the answer was good. True that there was no effect of the intervention. The reasons we might have seen a difference might be because our intervention arm looked just by chance higher, or that our control arm might have looked just by chance lower than the truth. But what we saw was in the control arm, the proportion tending was exactly what we had seen in all our formative data. So that gave me a bit of confidence that at least that half of the equation was probably, it's not caused by just a slightly lower attendance by chance control arm. The other thing is because our outcome was attendance within 4 weeks of the referral. At the point we stopped the trial, we have 4 further weeks of data to accrue. So that data is unbiased. Because we have these stopping rules, we've got this chance of stopping at a peak just by chance. The first few weeks of data, we might have just seen a big difference by chance. And because at that point we've gone over our threshold, we stopped the trial. But the 4 weeks of data afterwards that we've already enrolled, and we're just going to watch the results come in, that is unbiased data because we're not using that data to make a stopping rule. And when we analysed that data independently of the data at the start, we got exactly the same result. So that gave me confidence that the result. So while I wouldn't have put my confidence that the intervention arm was better than the control arm at 98%, which was the sort of posterior probability that we had calculated in conjunction with those other pieces of data, that the intervention was effective.
It's a great way to be skeptical of your findings and probe a little bit. And I get that, being on a team where folks maybe are more applied or ready to run with this, and you're the voice of caution saying, well, wait a second, let's just take a look at some more data, or at least you feel better about it. I love the study because it's such a great example of having a design and a method that is credible, but also fits the opportunity. And it's right for the moment. If you're doing a clinical trial of a new drug and you're looking at issues of safety, you wouldn't make the same choices. But for a text message based counseling, small add-on, and this might be one of 20 different small interventions you might want to try, tweaks to the program, tweaks to the delivery. You want to know quickly what things to keep, what to discard, and what a great example of how to do that. As I was preparing for today, I was reviewing your bio again, and I just impressed to see how many different types of studies that you've contributed to. And yes, I know this is the life of a statistician, but just looking down the list, I hope you don't mind if I just read for a second. Cluster randomized trials come out a bit, simulation studies, some diagnostic testing, population-based surveys, bibliometric analysis. You've really done most of the designs out there. And I'm just curious how your career has unfolded, how you decided to go into this work. You're at the London School of Hygiene and Tropical Medicine, and how you got there.
Yes. Jack of all trades and master of none, I suppose, a bit. So just about my background is I did an undergraduate degree in electronic engineering way back when. And then I went and worked for a telecoms company. So I was working in IT for a telecoms company. And so I did that till about 2011. So I graduated in 2004, worked for a telecoms company here in the UK in IT. And at that point, I got a bit bored of my career. And I realized what I was interested in doing. I was really interested in statistics. I'd always been interested in that. I used to listen to, I'd read pop science books and stuff in the news, epidemiology and that sort of work. So I decided to pursue a career change. So I went and did my master's here at LSHTM in 2011, just in my early 30s. And so came there and I stayed to do my PhD. So I enjoyed my year. I didn't intend to do a PhD. I thought, retrain as a statistician, go out into the world and not stay in academia. I stayed to do my PhD, which I did analysing some high dimensional data, metabolomic, heart disease. So that was quite methodological analysis. You mentioned some simulation studies, so I did a bit of simulation studies there. I would say I didn't enjoy the methodological work as much. And so as soon as I finished my PhD, I jumped into applied research. So I work for a group called the International Statistics and Epidemiology Group, LSHTM. And I found out I got a job, and half my job was working on a large population HIV test and treat trial based in South Africa and Zambia. So universal test and treatment. And half my job was supporting a wide range of studies in our eye health department. And that's how I got into these eye health studies. And so since then, I have carried on doing a lot of work with the eye health group across a wide range of different types of studies. So clinical trials, I would say, has been the majority of the work that I do work on. So right from the grant application design stage through to analysis at the end. But alongside that work, I did pick up bits and bobs of survey analysis, observational studies. I'd say that it's 70% of my work is trials and a lot of cluster randomized trials.
I love that you got an accidental PhD along the way.
I enjoyed being back learning, I think, and I wanted a bit more of it. And then when I was halfway through my PhD, I was like, not this much. But
every time I get to talk to students and earlier learners about these careers, I really try to emphasize that there's not really such a thing as a linear pathway. And yours certainly is one more example to add to the list of finding interests at different points in your career and running with it, and maybe even by accident getting a PhD, and then doing some really interesting work following.
I work with clinicians, and they're the people who come up with the ideas. I work with a guy called Matthew Burton, who's the head of the group here at LSHTN, and they're amazing visionary people who come up with lots of ideas of what we can do. And I'm not that guy, but what I think I'm good at is getting things to work. So how we can rigorously do the science. I don't have strong opinions on what science we should be doing, but I do have very strong opinions on how we can do science rigorously, and it should be done rigorously. And I feel that's my job as a statistician. I'm sure that's you feel too, Eric. We're often the sort of voice of making sure it's done properly.
But he's nobody's favorite team member, but hey, science is a team sport, and everybody has a contribution to make. And I really love this paper, and thank you so much for talking with me about it.